We just talked about DNA. We've already realized that there's a problem. Somehow we have to get instructions from the nucleus to the ribosome so that we can build proteins. Okay? First step to doing that is what we're going to learn about today. It's called transcription. That's how we copy DNA. Now, the copy of DNA is no longer called DNA. It's called RNA. And I know some of you have heard of that before because you guys were saying that when I was saying copy DNA, thinking of RNA. Alright, so just a reminder, DNA is instructions for making proteins. Proteins can be used in the cell or they can be used in the body, exported from the cell to be used by the body. A person, it's the you can see the muscles and tendons there. So here's the big picture. We have DNA. If we are just doing replication, we would make more DNA. But if we do transcription, we're going to make RNA. And then we do something else called translation. That's the second step that we're not going to learn about today. Translation will take RNA instructions and build a protein. That protein, of course, would give you your traits. Transcription is the process of taking the picture. The actual picture would be the RNA. Taking that picture and using it as instructions to build something.
Then what's the, like, you know how it's like transcription and translation? Then what's the protein to the trait?
The protein is the trait. So whatever proteins you have will be the traits that you show. Okay, so like if you have proteins for curly hair, your hair would show curly trait. Remember that transcription happens in the nucleus. DNA cannot leave the nucleus. You have to keep it in the nucleus. So copying DNA or taking a picture of it has to happen in the nucleus. Making the protein happens at the ribosome. What are proteins made out of? Amino acids. Building a protein means you're taking different amino acids and you're hooking them together, and they fold and they bend into whatever protein structure that you need. Transcription. Making RNA from DNA. Let me say this. Today we're going to be talking about making mRNA. However, transcription makes all types of RNA. Okay, so we're going to talk about making mRNA, but for future reference, you should know that there are other types of RNA, and they're all made by transcription. Alright, so making messenger RNA. Look down here at this picture. Do you see this strand right here? That blue strand is your DNA molecule. In the middle, I've got this pink square. That pink square is RNA polymerase. RNA polymerase is what type of molecule? It is an enzyme. You can see by the ACE at the end is your first clue. This enzyme unzips the DNA. See how it's been unwound here in the middle? That's the only way that you can read the nitrogen bases in the middle. You have to unzip DNA first. When the DNA is unzipped, RNA polymerase will then add mRNA nucleotides to start forming your copy to one side of the DNA strand. You don't copy both sides, not like replication. In transcription, you're just taking a picture of one side. Now, watch closely to my mouse. Your template strand is the strand that's being copied. This is my template strand. Is this actually being copied? This is a G, and what is this?
C.
C. This is a T. A. It's not a copy. What is a better word for that? It's a match. Okay, this is matched to that side. If this is the match to that side, is this identical to this or to this? To the top, right? So this is your coding, your template strand, which would make this your actual coding strand. This is the gene that we're going to use to build our protein. I know that you probably noticed right here, A is no longer attached to a T. When you're doing transcription and you're creating mRNA, you're no longer allowed to use T. Everywhere you thought you were going to put a T, you should now put a U instead. U stands for uracil, and it's the 5th nitrogen base. Okay, so there are actually 5 nitrogen bases: guanine, cytosine, thymine, adenine, and uracil. Couple of differences between DNA and RNA that you might want to notate somewhere. First of all, mRNA, or any type of RNA, you have tRNA, mRNA, rRNA, and some other types of RNA. Those are the 3 main ones. One of them is named tRNA. Guess what? There are no T's. No T's in tRNA. No T's in mRNA. No T's in rRNA. Are you seeing a pattern here?
Why not?
No T's in any type of RNA. And I'm saying that, and it sounds a little silly, but on your test, you'll see tRNA, and then you'll think it has a T in it when it doesn't. Okay, so no T's in RNA. And the other big difference between RNA and DNA is that RNA is just a single strand. See that green one? I'm not making 2 sides. I'm only making 1 strand. Single-stranded. That's nice because it's smaller, and therefore it can fit through a nuclear pore. We need it to be able to go through a nuclear pore and leave and go to a ribosome.
So does that mean that just make 2 copies and then send both of them together?
No, you're only copying 1 side. So right now I'm only copying my coding strand. I'm reading my template strand to copy my coding strand.
So you need both sides?
You do not need both sides to build that protein.
Okay.
Just need 1 side. Any questions on the job of mRNA or of RNA polymerase? Yes.
Which one's the template strand?
So your template strand is the one you read. The blue one, it's the one you're going to match. You're not copying it per se, because see right here I've got a T and up here I have an A. So it's a match because A matches with T. But because this one should be the same as this one is my coding strand. The top strand is your coding strand. It's the same as this one. See, C, C, A, A. Here, instead of a T, I have a U. Then I've got C, C, G, G. Make sense? 5' to 3'. I know I mentioned the ends. Remember that DNA has a direction. mRNA also has a direction. You build it from 5' to 3', which means you're reading the DNA strand from 3' to 5'. That's just something you have to memorize. Building your new mRNA from 5' to 3'. RNA polymerase only works in one direction. Okay. Steps of transcription. Number 1, RNA polymerase binds to a specific region on the DNA called a promoter region. Remember that you have extra DNA, and not all of your DNA codes for something. So here we have, in the picture, watch my mouse, right here I've got a green section here, a TA box, and a promoter region. Here is where special things are happening that are going to make my RNA polymerase attach here to start copying. This is an area that starts the trait, start of a gene, promoter region. RNA polymerase would come here, would open this up. Transcription factors are proteins that help RNA polymerase bind. Okay, that enzyme has these extra proteins that help it bind to your promoter region, and then once it's bound, it will start moving along and unwinding and copying. Step 2, once RNA polymerase is attached, so you've got your transcription factors, they help detach the enzyme to your DNA, you're in your promoter region. RNA polymerase moves along the DNA chain. It is assembling mRNA from 5' to 3'. It's moving on the DNA from 3 to 5, because remember they run opposite of each other. As it moves, the RNA polymerase is unwinding the DNA in order to read the nitrogen bases in the middle. If I'm unwinding DNA, what type of bond will I be breaking? Hydrogen bonds between A and T and C and G. You guys remember that? So once it's separated, free RNA nucleotides are added to the exposed DNA. So when the DNA is split, we're going to add RNA nucleotides to match it up. And like I showed you earlier, T matches with A matches with U, C matches with G, and G matches with C. We need to practice this for a second. Let's say I have A. Okay, if this is my DNA strand, and I was doing transcription to make mRNA, what would my complementary RNA strand be like? What would be the first letter here?
U.
U. Natalia, did you see that?
Yeah, the U.
My first letter here was a U.
Yeah.
Guys, you didn't change A to U. You didn't change A to U. You matched A to a U. I want to make that clear because when you start doing these worksheets, if you're saying I'm changing A to U, you're saying it incorrectly. You're matching it. You're creating the other side of the molecule. A matches with U. Who will match with T? A. You'll have a chance to practice this. I know that's your best wish right now.
Can't wait to practice transcription, Miss Rutledge.
Yes,
So
ma'am.
there'll never be like a T at the bottom.
Oh.
Maybe a top.
Good question. There will never be a T in the RNA strand. But don't think of it as the bottom. Because on the test, I could put this on the top and the DNA on the bottom. Okay? So the order doesn't matter. You have to pay attention to which one it is. And, guys, thanks for that question. You reminded me. I could ask you this question on the test. I could say, what would the DNA strand be that created that mRNA? And I'll give you the mRNA, and I'm going to ask you to go backwards. I want you to pay close attention. Because on the test, inevitably, this is what happens. You put T because you think of T changing to U. That's why I told you not to think of it as changing. Yes, Tavares? So what made this U was an A matching to a U. Yes?
So when the protein reads the mRNA, does it read the U, or is there a step in the process where the U switch to T?
The U's never switch back to T. U is what's on the chart, and it reads U. When the RNA polymerase, when the enzyme reaches the end of the gene, both the RNA and the DNA are let go of, and the DNA will recoil back into the double helix. Okay, the RNA is now just a single strand of letters, nitrogen bases that have been put together in the correct order. Those nitrogen bases have to be processed. What we just created was called an RNA transcript. You might remember exons and introns. I know I've mentioned these to you before. An exon is real information. Introns are extra letters, essentially DNA that you don't need. You have a lot of extra DNA. Eukaryotes especially have a lot of extra DNA. So before you can send an mRNA molecule out of the nucleus, you have to take care of all of the extra information. You only need your coding sections. The sections that actually code for some protein. That's all you want to send to the ribosome. You don't want to code for all this extra stuff that doesn't make any sense. So, first of all, you're going to need to cut out all the introns. See these introns in orange? You're going to need to splice these out. Exons you're going to keep. Introns you're going to cut out. Here's what that would look like. Your original copy, your original RNA is called your primary transcript. It is not a complete mRNA because it hasn't been processed. It's kind of like this, guys. You got your picture of your diamond. You're going to crop it probably, right? Maybe change the color, the hue, make it look good. You're going to process it, and then you're going to put it in some kind of package. You're not just going to stick a stamp on the front of the picture and send it. unless it was a postcard, I guess. But for most pictures, you would put them in an envelope and send them, right? So we need to process our RNA, crop it, change the color a little, make it look good, and then we're going to package it a little bit too. Yes?
How does it know which parts are introns or exons?
Enzymes. Has enzymes that go through and cut out all the introns. Alright, so here's your primary transcript, and then this would be your spliced mRNA down at the bottom if the green was all of the exons. Now you see there's no extra information. We can call this our mature mRNA transcript. This is our final picture. Okay, it's ready to go. Yes?
Say that you have one longer than brown eyes, or would it just be different?
It would be different letters, not necessarily longer or shorter. Some organisms have more DNA, but that doesn't necessarily make them more complex or their genes have more information. Okay. More post-transcriptional processing. Now we've got our picture already. We need to package it. Here comes the end part. Okay. We need to protect our mRNA on its trip from the nucleus to the ribosome or to the endoplasmic reticulum ribosomes. So we're going to put something on the end of it, okay? One side of it, the 5' side, gets a GTP cap. Now, a GTP cap is something you already know about, you just didn't know it was called that. Okay? Let me show you what I mean. If I have a sugar and a nitrogen base and a phosphate group, what is that called?
Nucleotide.
Good, it's a nucleotide. If this nitrogen base happened to be guanine, this would be a guanine nucleotide, right? If I added extra phosphates, how many phosphates would I have? 3. How do I say something for 3? Tri. Guanine triphosphate. Okay? GTP are guanine nucleotides that have extra phosphates. Why on earth would I want extra phosphates?
Energy.
For energy! Remember, the ATP, which by the way is this guy, adenine, right? Adenosine triphosphate is an adenine nucleotide with 2 extra phosphates. That's ATP. This is an adenine nucleotide. This is ATP with 2 extra phosphates. And remember we talked about how these bonds are really unstable between these second and third phosphates? They have a lot of energy. Okay? GTP cap. All I'm doing is adding guanine nucleotides that have extra phosphates. I'm going to add a bunch of those to the end of my mRNA. Now on this side, this is on my 5' side. You're gonna need to know that for your test. Okay? 5' side gets a GTP cap. The 3' side gets a 3' poly A tail. Guess what poly A is for?
Lots of A's.
Lots of A nucleotides. So that will be basically like packaging my picture. I've capped off both sides to protect it. I've given it some energy there in the bonds of my GTP cap. And this can be anywhere from 50 to 250 adenines. mRNA, that's what we learned about today. Transcription. Copying DNA to make mRNA.